Hello, and welcome to this e-seminar, which will provide an introduction to standard fish speciation and the Labelfish project. It is assumed that you have a working knowledge of Sanger sequencing and have viewed the previous e-seminar on DNA sequencing to support food labelling enforcement, which is available from the Food Authenticity Network at www.foodauthenticity.uk. A glossary of terms used in the e-seminar can be found at the end of the presentation. This e-seminar will introduce the analytical needs associated with fish speciation for food authenticity, the prevalent methods used in testing laboratories within the European Union, and the limitations of these methodologies. The Labelfish project will be introduced. A step-by-step guide to following the standard operating procedure produced by Labelfish, and an overview of the resources available for understanding and troubleshooting this procedure will be provided. Mislabelling of food products has been a priority issue since the prominent European horsemeat incident in 2013. In the wake of the incident, many sectors in the food industry were scrutinised, and seafood has been found to have a high level of mislabelling in the EU. As EU legislation does not define a standard methodology for fish speciation, a multitude of analytical techniques are currently used in seafood testing laboratories across the EU. EU-wide standardisation could further discourage and help identify fraud, resulting in improved identification of mislabeled fish. Harmonisation of methodologies has only occurred at a national level, indicating a requirement for an EU-wide standard operating procedure. The establishment of a multinational network could improve industry connections and coordinate a response to seafood mislabelling. The Labelfish project was started with an aim to address the analytical requirements in fish traceability. A survey by the Labelfish project identified the three most prevalent fish speciation testing methodologies in the EU. In PCR-RFLP, the polymerase chain reaction is used to amplify a specific fragment of DNA, which is then cut using restriction enzymes. The resulting fragments will differ in size between species due to polymorphisms, and can be separated by gel electrophoresis to produce a specific and identifiable banding pattern. According to a survey conducted as part of the Labelfish project, this is the most commonly used method for fish speciation in UK testing laboratories. Isoelectric focusing, known as IEF, operates on a similar basis through the electrophoretic separation of proteins based on their isoelectric point, the pH at which a protein carries no charge. Different species will have different protein polymorphisms, allowing for identification. Forensically informative nucleotide sequencing approaches are highly specific, as they rely on the DNA sequence of the sample. PCR is used to amplify a specific DNA fragment, followed by Sanger sequencing and comparison to a reference database. In 2010, the Food Standards Agency produced a standard operating procedure for fish speciation using PCR-RFLP with the Agilent 2100 bioanalyzer lab-on-a-chip instrument. As a result, the majority of public analyst laboratories in the UK and Republic of Ireland can use this SOP, a successful example of method standardisation supporting food labelling enforcement. However, across Europe, FIN's approaches are widespread, highlighting the requirement for EU-wide harmonisation of standard testing methodologies. The various methodologies currently available for fish speciation are suited to different types of analyses, and thus have differing limitations. PCR RFLP is a fast and inexpensive method, but may give incorrect identifications, especially with tuna species. A report by the Labelfish project suggested that the success rate of this method could be improved upon. Unknown species may also be problematic, but the method has some applicability to food samples containing mixed fish species. IEF is a protein-based method and so is incompatible with processed samples due to denaturation of proteins. FINS approaches are highly specific but are incompatible with samples containing multiple species. These approaches also rely on a high-quality reference database for identification. With the advent of easy and affordable sequencing, many such databases exist. Public databases are comprehensive, but may contain incorrectly labelled sequences, which may require additional interpretation. The Labelfish project, or the Atlantic Network on Genetic Control of Fish and Seafood Labelling and Traceability, aimed to address the analytical needs in fish traceability by reviewing levels of seafood mislabelling, standardizing identification methods, and establishing a network of experts and interested parties in fish traceability. The project ran from 2012 to 2015, with partners and stakeholders across the Atlantic area, and was funded by the European Regional Development Fund. The Labelfish website is still active and contains key resources and publications from the project, including the Standard Operating Procedure for Fish Species Identification published in 2014. As part of the Labelfish project, the current methodologies in fish speciation were compared and assessed for use as a standard method. FINS approaches were found to perform best across a variety of fish species and so were selected for further study. A plethora of FINS methods are available using a variety of target sequences. Additionally, different primers and probes are available for the same target. A barcoding approach utilizing the CO1 target, part of the cytochrome C oxidase 1 gene, was chosen as the universal methodology for fish speciation, and primers are specified in the Labelfish SOP. DNA barcoding functions by amplifying and sequencing a short standard part of the genome, the barcode, followed by comparison with recorded barcodes from known species. The CO1 gene has a high level of variation, allowing analysts to distinguish between closely related species. The SOP, developed as part of the project, utilizes BOLD, the Barcode of Life database, and is available to download from the Labelfish website. The first step of the Labelfish SOP is sample preparation. The protocol has been demonstrated successfully with fresh, salted and frozen samples, as well as some cooked samples, over a broad range of fish species. The SOP may not be appropriate for highly processed foods or products containing multiple species. Additionally, some samples may only be identified at a genus level. In particular, tuna of the genus Tunus have been found difficult to differentiate. Once samples have been collected, they should be immediately frozen and stored until use. When taking subsamples, the external layer of product should be removed using sterile equipment, and internal subsamples should be taken. For DNA extraction, the Labelfish SOP specifies a kit and protocol. All extraction batches should contain an extraction control, which contains no sample and is subjected to exactly the same processes as the other samples. Once DNA has been extracted, it should be quantified. The NanoDrop 2000 spectrophotometer provides quantification as well as an indication of quality, although other methods are equally valid. Regardless of the quantification method, the extraction control should contain no measurable DNA. Otherwise, DNA extraction should be repeated as contamination may be present. For the PCR amplification step, 4 primers are specified for the amplification of the CO1 target. These can be purchased from any oligonucleotide supplier. 3 examples of suppliers are shown on the slide. For LGC Biosearch, the primers should be ordered from the Custom Oligos section of the website. For Eurofins Genomics, the primers should be ordered from the Custom DNA Oligo section. For Sigma Aldrich, the primers should be ordered as DNA oligos in tubes. When the primers arrive, they should be made up to a concentration of 10 micromolar. For the reaction mix, it is recommended to prepare a mix for all reactions with a 10% excess. The Labelfish SOP specifies the use of Biotac-Tac polymerase, which arrives with reaction buffer and magnesium chloride. A negative control containing only water should be included, as well as a positive control containing a fish sample of known origin. The cycling conditions are specified in the SOP. Once amplified, samples should be stored at 4 degrees in the short term or -20 in the long term. After amplification, a check is performed to ensure the PCR was successful and the products are free of contamination. The Labelfish SOP provides a general protocol using gel electrophoresis to verify PCR success, but there are many alternative methods capable of PCR amplicon analysis. The Flash Gel system from Lonza and the Bioanalyzer system from Agilent offer quick and simple methods for checking PCR amplicons. If the PCR was successful, the positive control and samples should contain a fragment at roughly 700 base pairs, and the negative control should contain no DNA. If the negative control does contain DNA, the PCR should be repeated as there may be a contamination issue. A copy of the output, for example a gel photograph, should be retained for records. The next step of the Labelfish SOP is Sanger sequencing. The protocol recommends sending the PCR products to an external company for cleanup and sequencing. Any service providers may be used, and 3 examples are shown on the slide. Different providers will have different requirements for PCR product and primer concentrations. It is important to check this when selecting a provider. The 2 sequencing primers required are specified in the Labelfish SOP. 2 sequencing reactions are required, 1 forward and 1 reverse. This should be made clear to the service provider. Before sending the PCR products, it is important to ensure that all DNA is provided at the specified concentration and amount, and everything is clearly labelled. Once the PCR products have been sequenced, the raw data can be processed and a consensus sequence can be generated. It is assumed that the viewer has a working knowledge of Sanger sequencing. A previous e-seminar from LGC on DNA sequencing to support food labelling enforcement provides detailed guidance on the process of Sanger sequencing and the associated data processing and analysis, including troubleshooting guidance. In addition, the Labelfish SOP provides a detailed step-by-step guide to this process. Once the consensus sequence has been generated and the primer sequence is removed, the barcode is ready for species identification. The final step of the Labelfish SOP is species identification. This is achieved using the Barcode of Life data system, known as BOLD, a publicly available database containing barcode sequences for many species. BOLD is currently at version 4 and can be accessed at http://boldsystems.org. To begin the species identification process, the Identification tab at the top of the site should be selected. On the new page, the Animal Identification Database should be selected. This contains CO1 barcodes for various animal species. The species-level barcode records should then be selected. These are CO1 barcodes with a recorded species identification. Finally, the barcode generated in the Labelfish SOP can be input into the box at the bottom of the page, and the sequence can be submitted. Once the sequence has been submitted, the identification engine will process the barcode for a short time and then produce a result summary. The summary provides the sequence with the highest similarity provided, as well as the percentage similarity score. Additionally, there is a plain English explanation with clear wording to report the result, and a comprehensive analysis to look over if required. Ideally, BOLD will be able to provide a species match. In some cases, however, BOLD will be unable to provide a species match, but may identify the genus. In this case, BOLD will list the possible species or provide the closest species match. In some cases, BOLD will be unable to provide a match at all. The recommendation from BOLD is to check the sequence orientation. BOLD can also be used to search a wider database, for example the Public Record Barcode Database or All Barcode Records on BOLD. A different database could also be considered. GenBank from the National Centre for Biotechnology Information contains a vast array of public barcode records and can be searched by selecting the Blast Sequence on GenBank button from BOLD. In order to document the identity data from BOLD, the following should be recorded: a screenshot of the result summary from the BOLD identification engine, a copy of the tree identification diagram, which can easily be downloaded from BOLD. There are plenty of resources available online to assist with the process of fish speciation and usage of the Labelfish SOP. BOLD is the publicly available database used in the Labelfish SOP. It is straightforward and can be used without training and can provide results in plain English. Additionally, BOLD has a page with links to online resources, including a Frequently Asked Questions page and the BOLD Handbook, which provides further guidance on using the BOLD system. GenBank from NCBI is a collation of all publicly available DNA sequencing, including CO1 barcodes. However, there are many non-validated records present on the database, so identifications from GenBank may be questionable. The Labelfish SOP document provides a detailed demonstration of the protocol, as well as advice for troubleshooting the process. It is available to download from the Labelfish website. The newly published standard CEN/TS 17303 2019-06 provides standardized methods for the DNA barcoding of fish using the CO1 and Site B targets. It is available for purchase. The work from the Labelfish project had an immediate impact on mislabeling rates of European seafood, which was reviewed in 2015 upon completion of the project. Mislabeling rates were found to have decreased, a possible response to the policy changes and method standardization brought about by Labelfish. Longer term, the Labelfish work has been continued in the Sea Traces project, which aims to build upon the foundations developed by Labelfish. Some key aims are to produce an SOP for the identification of mollusks and crustaceans, to improve the robustness of tuna identification methods which remain an issue, and to explore portable diagnostic methods and mixed product analysis. The Sea Traces project will run until 2021. To briefly summarise some key points. Seafood mislabelling is widespread in the EU, and EU legislation does not specify a standard method for fish speciation. The Food Standards Agency provided and disseminated a standard method for fish speciation based on PCR RFLP. This was adopted by public analysts in the UK and Republic of Ireland, but is limited and non-specific. The Labelfish project aimed to develop an EU-wide SOP for fish speciation. The SOP developed as part of the Labelfish project is a highly specific FINS approach, which utilises CO1 barcoding and the public database BOLD. The work of the Labelfish project is continuing as part of the Sea Traces project, which aims to further improve standard methods in seafood speciation. We would like to acknowledge the support received in preparing, delivering and funding this e-seminar, in particular the Department for Environment, Food and Rural Affairs, the Government Chemist Programme for 2017-2020, the Department for Business, Energy and Industrial Strategy, the Food Standards Agency and Food Standards Scotland. And finally, we would like to thank you for your attention.